Okay? Hmm? Okay. So, welcome everyone. It's so hot. Okay, yay. I really need it actually. I want to start with the announcement. We're getting toward the end of the semester, and I don't know if you remember, but the grade is also built. There is a project that you need to hand in the end, or to describe here. So Shai will announce it through the forum, maybe after the class today or tomorrow. But there is list of topics. All are methods which are related to what we learned, but none of them are actually methods that we learned. And so the thought is that you will create groups of 2, select a topic. I think it's first come, first serve, right? And then you will choose so you will choose this topic. You will then make a talk. There is a explanation that we'll send, but the idea that you will first present the method, relate it maybe to what we discussed until now, the advantages, disadvantages, and show and kind of present an article that was using it. Altogether should be a very short talk, so it's going to be kind of like, so each group of 2 will present. We're going to do it somewhere in the exam. We kind of like reserve time in the exam. There is exam time. The exam is actually this seminar that you all going to present your presentations. So, well, that's just the time to select a topic and start thinking about that. And when you think about that, and when you're selecting the article you want to present, or maybe the material you want to rely on when you do your work, you're very welcome to come and discuss it with us.
How long exactly each presentation?
15 minutes? 15 minutes. Still, I don't, how many are we? 20? Less than 20. It's going to be a long day, a presentation. No. If you feel like you want to do analysis, if someone want to do something else, particularly if it goes with data, you should let me know and I might agree.
Just present the technique that we...
Present technique and show how it's been used, kind of also contrast it to what we been presented, so advantage, disadvantage, the physical sources, things like that. So it's essentially another class, kind of like a blitz class of many methods. You will all help me present. Okay, today we shifting gear from anatomy. So we did anatomy in the last 3 classes using MRI, and today we're gonna move toward fMRI, which most people who use MRI actually use it for fMRI, at least in brain research. And what we will do today is we would look on a little bit on the methodological aspect, what exactly been measured, how we measure it, and then we will try to look on the neuronal source, so why we say that something that we measure with MRI relates to neuronal activity. So the topic is the neurovascular coupling. That's how it's called. And so let's start. And before I'm starting, just one more announcement. Just after this class, there is a seminar from Ariel Rokem, which is coming from Washington University, and is actually expert in MRI in general, in fMRI in particular. with both. He did a lot of vision research as well, and he's gonna talk about big data analysis using these huge corpuses and how can we use them, this type of fMRI data to learn about brain activation and brain systems. So you're very welcome to join and essentially stay. Okay. So fMRI. Maybe some of you saw this image, and this is a typical retinotopic mapping kind of stimuli that the subject look at, so they fixate on this image, and there is this wedge that change in their size. And when we look on activation with fMRI, we would see that we can see this. So this is a movie kind of summarizing fMRI activation, and we can see the rings of activation of eccentricity along V1 here, and the other visual cortex around it. And that's what I'm trying to do here, is to convince you that it's actually amazing and very useful. It's amazing that we can, with this magneting, and you would hear where the signal is coming from. It's kind of like, really? And we can measure that and measure, for instance, change in area that react to different eccentricity in the stimuli. It's pretty amazing, but we can, and we do it very well. And Yuval, in the next two classes, will tell you more about the analysis and how we get to this kind of images and this type of analysis. And one of the topics, by the way, is population receptive field, which how you map those the population of receptive fields in the visual cortex, for instance. Just, I mean, topics for presenting in the end, your presentation. Okay, so it's a very nice tool. It's nice, and everybody tend to use it because essentially we can measure brain activation without opening the skull, right? That's a big thing. It allows us to map brain regions, for instance, again in the visual cortex, and just give you an example of the resolution and type of analysis that, in the end, we kind of. I'm trying to give you motivation here of why we should care. And so, for instance, using this stimuli and activation that we see along the visual cortex, we can then map a different visual brain area. It's not just like in this image we can say, okay, there is activation, some occipital activation that relate to visual scene. In here, people using stimuli like the one we saw and others, which is like rotating wedge, you change the polar angle, and we can actually map different brain regions like V1, V2, V3, and others. Actually, now about probably more than 20 regions that are now identified in the occipital cortex, for example, cerebral fovea and so on. It's a really rich data set and information that we can get. So, who remember what is the source of all of that? What are we measuring actually? In the fMRI, what is the source?
The magnetic field by the oxidative hemoglobin, right, is the same tissue around it. When it's deoxygenated, we get disturbed magnetic field. That's what we try to measure, how much disturbed the image.
Right. So some kind of if you want, artifact or some kind of inconsistency in the main magnetic field due to the presence of or the ratio between oxy and the deoxyhemoglobin. The deoxyhemoglobin is paramagnetic, and it creates this if you want, artifact in the environment, which we can measure, right? It creates a reduction in the signal. Okay, so this is basically what we said, and it's important in here. It's not exactly what we said actually. What we can see here, and that's what we're going to get in toward the end, is that we're measuring something that has to do with blood vessels in the cortical area that we're measuring from. Okay, so we're not exactly measuring neuronal activity. We're measuring something that has to do with the oxy and deoxyhemoglobin, that has to do with the structure or the machinery of blood or metabolite arriving to blood and metabolite that arriving to the area of activation. Okay, so that's the source of the signal, and these changes with function will create a list of changes which we will go over, like changing in the metabolism of oxygen, changing cerebral blood volume, and in the end, what we call BOLD, the blood oxygenation level dependency signal. And actually, this and it has to do with activation, yes, but also, and it's actually monitor and highly related to the effect of astrocytes. So astrocytes are big factors in modulate and control those effects of those changes that drive the BOLD signal. Interestingly, and I'm not going to speak about it too much, is another important method that allow us to measure function in the brain without opening the brain, although I wouldn't call it non-invasive in the same way, called PET. So in PET, we inject radioactive materials, and we can see the consumption of them, or we can see the emission coming from these radioactive materials, and we can kind of localize where it is. And so one of the options that people use is either using oxygen, which is radioactive, or glucose, which is radioactive. And so in a way, there is some relationship between what is measured with BOLD, with fMRI, and what is measured with PET when it's a functional measurement. In many ways, it's more specific in PET. You can actually target the glucose. On the other end I mean, nobody really wants to be injected with radioactive materials if it doesn't have to. So it's not as, well, people use it. But for instance, Shira Tzilda just arrived to the psychology department here in LSEG using that to measure changes in mothers after birth and so on. So there is people actually using it for research even, not only for medical use. For medical use, everybody using it all the time. But still, the other effect that relate to fMRI is much more used because we don't need to do any invasive effect. Okay, so the idea that there is a change in the blood, essentially in metabolism that is related to function is really, really early. Here is from the work of Foray and Sherrington that you might heard about in other classes, right? And already here in the late 19th century, they're talking about changes in cerebral metabolism that relate to function and activity. In this time they wouldn't know to say that it's the neurons activation and so on, but this idea is really early. And in the beginning of the 20th century, there were several works that tried to measure it, and I will show you some examples that are kind of funny. This is a review that went back to one of those work of a guy named Angelo Messo, which is famous for another work which I show you, but they actually found that he had two very famous methods. So one is more famous than the other. So here they went to the other one and say, oh, that's been forgotten, but it's actually very cool. It's actually almost everything that he's dealt with is very similar to fMRI. Besides what he did, so they actually built again his machine and so on. What he's measuring is measuring the weight of the brain during activity. So what he did, he built. This is his drawing. He built this weight when people were lying on it, and then when the brain were active, he would try to measure some changes in their I guess volume of blood. And well, when there was some kind of a task. I actually don't know if that was very useful. He had another method which was more useful, but they rebuilt it. I don't remember if they were saying that in the end they were able to actually measure it or not. But just showing you that was something that people were doing. He was actually working with people which have very, very thin skull. And in this case, he actually can. So in this subject, you can find them. They're rare, but exist. You can actually look at on their blood vessels, and he was drawing them. What he was seeing, this fluctuation, and so he was measuring the cerebral blood flow essentially. And here is his description where Mr. Ken was resting peacefully when I said I just a few expressing the impression that his wife had made upon me when I first saw her. Ken did not speak. The blood and event increased immediately, and da da da. And so he was measuring that very, very early. Okay, so that's interesting. And just another anecdote for you. Another one or another two? Yeah, another one. So this guy, Fulton, I think working with mostly with soldiers in World War I, but in this case it's a different story. There was this guy that whenever he was seeing something, they would hear noise from the back of his brain. Something. There was just constant noise, and when he was closing his eyes, he wouldn't anymore. And that was kind of said in here. And from that, they assume there is something that has to do with brain activation in the occipital cortex that has to do with vision, and have to do with change in blood, arriving or flowing to this region. And this is the description of what happened afterwards. When they open it, they do some kind of dissection, and there was a big cloth, something like that, that was there. You remove that, and then the noise moved, and also his vision was better. Anyway. Okay, we're going back to here. So, in modern days, we're not using this method anymore. We try to use other methods, and we mostly relying on this change in the oxyhemoglobin, as you said, and their effect on the magnetic field. And so what we would like to measure in the end is what we call BOLD, the blood oxygenation level dependency. And the thought is that in the veins, so while oxygenated blood arriving to the area, there is consumption of oxygen at the region of activation, and that will change the ratio of oxygen to the oxyhemoglobin in the veins, the area that takes the blood, the used blood, and that will change the signal that we measure. And note that we'll see it in a second. There will be actually greater increase afterwards of oxyhemoglobin that arriving, actually greater than any change that we'll actually experience in terms of the oxyhemoglobin. So the main effect will be actually opposite effect. But actually, we're going to have more oxyhemoglobin and not less deoxyhemoglobin. Both of them will occur. This effect, the other one, arriving of a greater amount of oxyhemoglobin, will be the one that mainly manifests in our measurement. Okay, so what type of measurement in MRI is sensitive to that? So the ratio of oxyhemoglobin, you already told me it has to do with the change in the magnetic field in homogeneity. So what measurement in MRI care about that and will allow us to measure something like that? Any thoughts? That goes back to our first class about relaxation, and I told you that I'm telling you all those stories and they will be relevant for something. So I told you a little bit about T1 before, about some relaxations, but there is other relaxation where I said that will be relevant for fMRI. So do you remember? You can try, you can make mistakes, it's fine. T2 star, right? T2 star. And do you remember what is T2 star? You're right, it's T2 star. So T2 is what? Is when you tilt the vector toward the XY plane, and that's the 90.
Yeah, that's the 90, and then the 180.
Okay. So the 180 is more is our method to get the T1 effect in many cases, but there was something about the vector that was on the XY plane, that the vector we actually measure. There was something about it. It was the transverse relaxation. So that's the decay of the signal, and we were saying that the transverse relaxation has two sources of. So what do we mean by transverse relaxation first? Do you remember? Yeah. So they're losing phase. In the beginning, they're all in phase, right? They all point one direction, and then we can actually measure them. And as time passes, they're losing phase. Essentially, they sum up to 0 in the end because they are not pointing to the same position in XY plane. So we said this dephasing actually happened even faster than the main T1 relaxation. So there's a loss of signal due to this dephasing, and we call it T2. And then, as been already mentioned, there was something on top of this T2. We call it T2 star, and that was what? Who remember? Yes. So it was any inhomogeneity which is not coming from spin-spin interaction. So it's coming from the environment. If there's any inhomogeneity in the environment, that will cause dephasing. And we actually said that in general, as long as it's there, we can also do the 180 and kind of like undo this effect. That was with the 180 maybe. So there's this T2 star, which is sensitive. So we're first talking about dephasing. You remember this movie? There was this vector in the XY plane, and then as the spin were dephasing, the signal was lost, and we could find a decay in the signal, which was about an exponent. It looked like an exponent, and we gave it, and we call it T2. And then we said that there are two sources for this decay. One of them, so this loss of homogeneity could come from spin-spin interaction. So that means that I'm a magnet and you're a magnet, and each of that creates some inhomogeneity or some change in the magnetic field that I'm experiencing, and it's different between us, and so we're not aligned anymore, right? As time passes, we aggregate more of this effect. So this is the T2, and the other one, the T2 tag, which together sum to T2 star, is everything in the environment that affecting this decay. And so the deoxyhemoglobin in the blood is something like that. It creates this inhomogeneity in the environment. And so if we will have a measurement which is sensitive or weighted by T2 star, we would have a measurement that is sensitive to the oxy and deoxyhemoglobin in the blood or in the environment. Particularly if there will be changes in that, we might see that. Well, the effect is actually T2 tag. There is way for us, but we never do it, to separate the T2 versus T2 tag. So we're measuring the join of those, you sum it in the 1 over, so in the R1 if you're in the rate. So we typically talk about T2 star, which is the joint effect. It's always the case that this one is much faster, so it's also dominant. And the other thing is that we don't expect that those effects, that affect T2, change as function of activity, but this one would. So we actually, in reality, you would see that it's actually rare that you're actually measuring T2 star in our functional measurement. We're just going to do a measurement which is sensitive to T2 star. We're just going to do it twice, one on, one off. We do many, but in general, there will be a contrast of with activation, without activation, with the same measurement, supposed to measure the same T2 star effect. And if there is a difference, then we would assume it's due to some changes in the oxy and deoxyhemoglobin, and then neuronal activity. People will call it neurocorrelates. When they say neurocorrelates, they actually say all those things that you know. I want you to know, actually, because there's people who are using fMRI, they have no idea. But it's important that you remember where the signal is coming from. Okay? Okay, yes, we said that. So just to remind you, and not exactly to remind you, because actually there was a slide that I skipped when we were talking about gradient echo, or about sequence sense. So there is gradient echo, we get to that when we get to fMRI. So here it is. So this sequence is an interesting sequence in the sense that we start with a 90, and then signals are supposed to decay. Remember, there's supposed to be this oscillation, and signals are supposed to decay. We're doing something funny. We're adding a gradient that will create the signal to decay even faster, really crashing the signal. And then we're using the same gradient but in the opposite direction, which in the end creating an echo. And so this echo allows us to get a peak here, like somewhere in the time of the decay, while allow us also to encode the space. I don't know if you remember that, but I was mentioning that we have to use gradients to encode the image, right? So we're not measuring from one voxel, we're measuring from many voxels. So we have to use gradients. So here we use the fact that we're anyway going to use them, and we're just going to create an echo sometime along this decay. So we'll have a peak, we have a signal, and this will be our signal in fMRI. I put it? You put it. Thank you. So you put a video of gradient echo in. Great. Illustration. Okay. So we have a signal here called gradient echo that allow us to have a measurement which is sensitive to T2 star. Actually, the time that we'd use, the TE, time of echo, we change the weight of T2 star. If we wait too long, we're going to have nothing. We have no signal. If it will be too fast, we might not have a big effect of T2 star. The signal wouldn't decay enough. So selecting the right TE is critical, and people actually think about that when they're doing an fMRI experiment. The right TE will change the weight of the T2 star in your measurement. Okay, so here is the story. Here is how it looks. So this is the signal when we have a good activation, some good area which we can actually get a good signal. It's in green, and in red is when it's in rest, let's say in the visual cortex. So you see the effect is tiny. Tiny, 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 tiny. Fine? Just for you to know. And if there is a measurement that we can have, it's here somewhere in those TEs. Okay, this is the time that we can actually get a difference. So we can actually plot one against the other as function of TE, and we're going to get something like that. We're going to get a peak somewhere between 30 to 60 milliseconds. So that will be the typical echo time that is used in fMRI, where we have a maximal difference between those measurements, and still in a very good area, it's about 1% change. Okay, very small signal. And yet, it's possible to measure that. Who knows how we can overcome the fact that we have a very small signal and there is noise in the system? What should we do? Yeah, just average like hell. So we're doing many measurements, and which allow us. And there is different way to do this averaging, and maybe Yuval will discuss that next week. Next week, yes. Yuval is the next 2 weeks, and then you. Yeah. But it will be almost impossible to do it in one shot. So something that we aggregate and to get this map that you saw. Also, those maps that you saw with the fMRI that was moving, this eccentricity, this averaging of probably an hour of scanning, again and again, visualizing this like extending rings. So it's very boring experiment. Yeah.
There's 1% change is the change between the ratio of deoxy and oxyhemoglobin in the vein. Like what do you measure exactly?
You measure a change in your signal. You don't know what you measure. The only thing you know is that you measure something which is weighted by T2 star. Also weighted by other things, right? It's weighted by proton density. It's weighted by maybe by T1, by T2, but weighted by different MRI aspects. But also, and highly weighted by T2 star, you select here the echo time, which is there is the greatest weight in T2 star, giving a typical T2 star in the brain. Okay, and you measure it twice, once with a task, once without a task, and you do it many times, and then you use this kind of, you do an analysis kind of like separating your time points to the ones which are on versus the ones which are off. Typically the design is more richer than that. So I give you just kind of giving you the flavor of that, and then as you do that, you can ask where I find correlation between the design of my experiment, the on and off periods, versus the activation in the signal. And we just say, oh, here's the area that was kind of turned on. I see a change. What is this change? You don't really know. We're going to talk a little bit about it. But as you do the experiment, you want to imagine that it's always the same kind of principle. Neurovascular coupling, neurons activated, change. Is actually debate. I wouldn't say hot debate because most people kind of just. But some people care, and for them it's a hotter debate. It's in area like visual cortex is very early. It's easy. Like that's why the reason I showed you. It's easy to know that we know there is map in V1. It's like a function of eccentricity, for instance. We know it for monkeys. We know it for eco. We know it for many things. And then you can kind of see it again. We just say, okay, I'm actually measuring function. It's probably this change in oxydeoxyhemoglobin. In other areas, when people are doing something very sophisticated, some very high-level cognitive task, and they find a small change between this and that, they imagine it's the same. They're really just imagining it's the same, or they're relying on work of many others that show that it's sound. But that's always a drawback. Okay, so the very early measurement, and this is from a review of the review is actually from Camille Ogunville, which is like one of the great founders of the field, the head of one of the heads of the Human Connectome now, sitting in Minnesota. Really, one of the founding fathers. The work was of Ogawa, the early work, and he's telling us how they measure that. So it was done. Actually, it was done in competition between two groups. One of them was working in Bell Laboratory and in Minnesota, and the other one was in Harvard, and they were competing, and they was trying to use this bold signal in order to measure function. So that was in the early 90s. Actually, interesting story. They both met each other in the main conference of the field in 91 and discovered that both working on that. And both, he said here, both kind of realized they don't have enough. It took them another half a year to get to the level that they actually have a functional measurement. And Camille actually describing how they actually went to Nature and they were rejected because people thought it just nobody gonna care. And in the end, both groups published in 92 together. This is the work. This is the first figures. And so what do we see in here? So this is one way of looking at that. They have blocks of off and blocks of on. Okay. This is baseline of the signal, and then they do blocks of off, like several minutes of not showing anything, or maybe say to somebody close your eyes, or just properly. Maybe I don't know exactly if how careful they were. Now we would use some kind of a fixation and don't move your eyes and so on. And there won't be other stimuli. And then in the on, you would probably see something that is some checkerboard that just move. So you want to constantly activate your occipital cortex, and then you look on the activation between off and on and off and on. And in the visual cortex, in particular, these other areas, in the visual cortex, we have a very good signal, about 1 to 2% signal change that we can measure. We can see those changes. So this is one example. This is they actually use slightly different sequences. This was just an EPI, gradient echo. This one had an inversion. So there's also a weighting of T1 in this one. Never mind. What we use right now is this one. This gradient echo. Again, they look on. They took several measurements. They can show you that in measurement in area 3, for instance, which is in here, it's in the cerebellum. It's just a flat line. While in area number 1, which is exactly around the calculon area 1 and 2, they found this. When the light is on, they found activation and then reduction and activation and reduction, and fine. And so they are very excited. They're measuring function activation in the brain. Great. So then the next discussion was where this signal is coming from. There was a thought that it had to do with blood, but should we care about it? It's coming from neurons. It doesn't come from neurons. So that was kind of early work from Eager and Russ. Also, about 10 years after, where they actually measuring in monkeys fMRI and neuronal activity, so measuring kind of counting spikes, and they were finding this very strong agreement between the amount, so they were changing the contrast that the animal were seeing, and they were finding a very nice agreement between between essentially a number of spikes to level of activation in bold, and that people were very, very excited. Then there's a little bit of a hiccup that I will describe to you. But anyway, that was kind of that was a promising result. Later, there is a massive work from a group that back then was in Max Planck of Nikos Logotatis, and that was working also with monkeys, and he built like this this setting in which he can measure fMRI and record both multiple electrodes. Both LFP and multiple unit activity from the same location. Okay, so he built this ring that was very, very exciting and important. And so this is the signal is separated to LFPs and this multi-unit activity, and you know all about it, right? Because you learned that in the other class. So this one probably related more to spike and neural activity, right? And LFP, some kind of a sub-threshold electrical change in the tissue, which doesn't correspond exactly to spikes. It has to do related to spikes, but it's not exactly that. It's in a way really look more like our EEG recording, but under the skull, right? Or maybe like ECOG recording when we put just a electrode on the skull, okay, under the skull. Okay, so you know these definitions, right? And so what they did and what they measure, so they show a stimuli and they count spikes, for instance, and so with the stimuli here in green, and they found that there is agreement between them. There is also agreement with the local field potential, and also the bold is activated, and there is also delay, which we're going to discuss. The bold is delay, is in delay. It doesn't activate exactly when. So in terms of time resolution, we're going to discuss it. We're really not there, right? Time resolution of the bold activation seems to be delayed, but besides being delayed and continue much longer after, right? Both those things. So the time resolution here is really clear. It's not sharp to the stimuli. But besides that, it seems to be a very nice agreement in all of those things. But when they continued the experiment, they actually discovered that while as you increase the time of the stimuli, you might see a reduction in your multiple unit electrode recording, and you won't see a change in LFP, and the bold goes with the LFP in this case. So there was a better agreement between LFP and bold than between the multiple unit recording and bold. Okay. And here again, in terms of LFP, and this is a work that will come afterward. It's really interesting. People use this work to say this one thing and the opposite. They found that there is, as you increase the length, there is kind of linear relationship between the LFP activation and bold. People say that it's linear. Then there is also a break in that. So some people say it's proof that things are not linear. So it depends where you are. But I assume that for instance that Yuval will discuss it and will tell you that things are linear. They're pretty linear, but they're not fully linear. Anyway, they're linear with LFP. Just one more experiment that they did. They actually put a thing that blocks spiking. Okay, so they block spiking. So LFP doesn't change. So there is stimuli. LFP doesn't change. There are no spiking, and there is a bold response. What does it tell us? Who wanna?
The supply of energy that the neuron needs is not from spiking, but from the
Good. I was looking for this. So it's true it has to do with the supply of energy and so on, and we're going to discuss that in the next part after the break. About the supply of energy we're going to discuss soon. But it seems that the signal is also, at least because there is no spiking going out, so there is no output. But there is still this LFP change, there's also a ball change. It seems that what we're measuring in ball is at least heavily influenced also from the input. So it is not. So if one wants, and some people like Yossi, if you talk with him about it, Yossi Ram says, what is this thing? Who cares about that? You don't measure. It has nothing to do with spikes, and you're just wasting your time. And so with people who would actually concentrate on spikes, they kind of, I don't know what you're measuring. Stop. Still, well, we should be aware that there is at least a strong sensitivity to the inputs, not only to, and not always to the output. Okay, let's stop here and take a break. שי, אולי ניתן איזה קירור ככה בינתיים? אני אדליק מזגן קצת? שיהיה קריר, לפחות ההתחלה.
כל הדברים האלה, אני לומד בימים האלה. מטעם של השנה.
so we ended up. What? Okay. Okay. We ended up with this question of where the signal is exactly coming from. There are recent work that try to use optogenetics to try to localize the source of the signal. And this is work that came from the Zeisler lab, and Inbal is one of the authors on this work, in which they use optogenetics, activate neurons, and then find bold activation. And it was for a moment they said it's a proof of who is actually driving the signal. That's what they showed there. And then there was Nicholas Logothetis, the guy that did the work. It's a funny button that you can read, which they comment to each other, essentially saying that also with optogenetics it's really hard to know what exactly. I mean, you're targeting, but then the signal is. It's not control of what exactly been activated and where the bold effect came from and so on. So I don't think it's been resolved using this work, but it got some attention for some time. There is a recent work that was presented here in LSEC by David Kleinfeld recently, in which they use calcium imaging and compare it to bold activation. And in there, in particular, they look. There is some interesting. One of the topics that are there in the topics that you can choose is something called resting state fMRI, which have to do with slow oscillations that people find in the bold signal. And so they try to see if they can match and find something which like that in the neurons. Are the neurons also have this very slow oscillation? And they're suggesting in this work that there is, if they look on calcium, a very slow calcium oscillation within the cell, a very slow change in calcium, they find some relationship to the bold effect, and they relate that, I think, to something called smooth muscles, which sit on the blood vessel and have there some kind of some oscillation of their own, which slow oscillation. Maybe this drive both the calcium and the bold. Anyway, that's just to show you this is from this year. All this topic is still very active. So where exactly is the source? Or everything is an neuronal source? Maybe not. I'll just mention one more work that's coming from I think last year, from John Wiener, who were here actually a few weeks ago in the ENU day, in which they tried to model the bold signal and compare it to LFP signal in ECOG recording. And the interesting thing is that they kind of generating one model of activation of neurons, in this case in visual area, and try to kind of and build predictors of what will be the signal, what will be then the effect on the bold, and what will be the effect on LFP. And the model works very nice. You can predict one signal from the other using this framework. So it's interesting. It's not a proof, but it's interesting. I think conceptually it's interesting because they're putting things in one framework, and they can make predictions, which is also a good sign for a model, a quantitative model. You can, for instance, show that gamma oscillation cannot be less related to bold versus the broadband. And slower oscillation could be better matched to the bold effect, and so on. So, and maybe you remember that when we talk about EEG, there was different bands, right? Different oscillations that been measured. So here they kind of allow us maybe to relate one to the other with this model. Okay. Yeah, that's prediction and so on. So let's talk a little bit about the blood supply to the brain. And so this is just, I think, a brain filled with plastic to just show you the how vast and big is its blood vessel system in the brain. You probably know about 15 to 20% of the total cardiac output goes to the brain. So the brain is very well. It's an organ that wastes tons of energy. Okay, it's like about 20% of what the heart drive goes to the brain. Although in terms of weight, you know what is the weight of a brain, of a human brain, about?
1 kilogram?
Yeah, about 1.5 kilograms. So it's definitely not the amount of energy that is consumed by the brain, or the amount of blood that arrives to the brain, is not in relation to the size of the brain, weight. And we can find variability or change in the way the blood is allocated in the brain into different regions. That's the key, right? That's key for our measurement. If everything was just either you get a lot or get less, depend on your activity, but it won't be specific, then we won't have. There is some specificity in the way this blood is circulated or allocated per region according to their activation. So there is some flexibility in the system to allocate the blood according to need. So terms like arteries that derive the oxygen, the glucose, and other metabolites, right? Those go to small capillaries in the end. That's what actually get really close to neurons. I think each neuron would have a capillary. What is the distance? Around 100 micron, something like that. So there is good proximity for each neuron to be around the capillary. Not one-to-one, for sure not one-to-one. What does it mean that it's not one-to-one? It's just a priori about our resolution in fMRI. Yeah, by definition, it won't be neuronal resolution because we don't have a capillary per neuron. So even if we would able to measure that resolution, but the system doesn't work like that. There's limit in there. And then there are veins that collecting that collect the blood, CO2, and so on. Because the brain is such a vascular organ, there's also a great risk for damage, right? And any kind of problems in the blood vessel. You probably know about stroke, right? That's the most known defect of blood vessels, which is massive, right? A stroke will have a massive functional effect on the brain and cognitive and so on. There's also a great change in terms of in aging in blood vessels. I will just show you that in a second. Blood vessel distribution change, and also change in different other disorders, and that's actually important for us. I want to talk about it later. Okay, so I'm not gonna do with you the mapping of blood vessels, although knowing where are blood vessels, and especially where this huge one, the huge sinuses, is actually critical. Actually critical because they have such a great effect on the ball signal that we actually cannot measure there anymore. Okay, so if one ignores that and see they show an effect, don't see effect, the effect might be flipped. Everything is kind of like when you are really close to a big sinus or big blood vessels, you're essentially measuring something very different in your blood-related measurement. Okay, in your ball signal. So your ball signal is not actually what you think it is. So some consideration people should, but not always consider. I will show you an example for that if I will have time. So this is a distribution of blood vessels along here again along the occipital cortex as function of layers, and we can see also patterns of blood vessels as function of layers. And if I really get to what I want to say toward the end, I will show you that people try to not measure that with fMRI. But there is a pattern of blood vessels as function of layers, and also as this is building one like that. Actually, if you were in the talk a few weeks ago of Pablo, that's what he's doing, right? That he's looking on this blood of the distribution of blood vessels in in the cortex in mice. That's the research. That was kind of what he was doing, and the fMRI people in the audience were because this is actually relevant to our measurement. So that's to put the things in context. We can see blood vessel distribution change between regions. This is a very famous one between V1 and V2. But in general, there is a difference in situ architectonic organization. That means cell density between regions, right, in the brain. We can also see a change in terms of blood vessel distribution. But also important to remember, if you're looking on fMRI, comparison between regions might always also relate to that. Okay, I mentioned that before, function of age. There is a huge effect in aging of blood vessel distribution. That's what we see in here. And the fact that there is a change in blood vessel distribution in age, again, make it hard to make comparison if you don't take it into account. It's really hard to take it into account. Okay. More numbers about the system. So this is the mean cerebral blood flow, about 60 ml per gram per minute. It's allocated to be about 21% in the arteries, about 33% in the capillaries, about 48% in the veins. What else that were interesting in here? All the blood vessels together in the brain take something about 4% of the volume. That's how much it is. There is a great difference between gray and white matter. It's much more. Where is it? It's here. The cerebral blood flow to the gray matter is much greater than in the white matter. What does it mean for our measurement? If you have much less blood vessels in your tissue, sensitivity will be lower in the white matter. And actually, it's really, you don't see it. So kind of all these bold measurements are always a measurement in cortex mostly. It's actually also very hard also to measure in subcortical regions. And there is no measurement in the white matter. Why not? It's not like there is no use or metabolism in the white matter, just much less. So really getting to the floor of the SNR, and we don't really see, people typically don't look at activation in the white matter just because there is no signal, almost no signal. Although there is few groups, really few, I think less handful, that consistently say that they are measuring functional measurement of BOLD in the white matter. But it's kind of like a, there is still big there's uncertainty about those measurements still. Okay, please.
So in young children that they have less elevation,
Uh-huh.
so would be more signal.
Good question. In the white matter? You mean if there is more activation in kids in general?
Yes, there is, I know. Right?
Depends, actually.
Okay.
Depends.
But generally,
There are differences between function of age. So also for young for young children versus adults, and if it's due to changing in blood vessels, I don't know. I don't know how big is the change in blood vessels. So in aging, it's a different effect, right? It's just you're losing your vessels. I don't know where blood vessels stop developing, or where you're getting kind of like your almost mature blood vessels. It might be very early. You might assume it's actually pretty early, but I don't want to say things I'm not certain about. It might affect. Also, there is a question of efficiency of usage, and that might also change as function of age. There's a difference in specificity for sure between adults and kids. So there is more. People sometimes see more activation in terms of space. There are more activation, and then people would claim that has to do with area become more specified, more specific to their job as you develop. You actually been more efficient in kind of allocating the right resource to solve the task. That's a hand waving, what I just said. Okay, so to relate neuronal activity to change in blood oxygenation, I didn't do that yet. There were several interesting works. Most of them were done in the cerebellum, actually. And in there they use, and actually was also described here in the ENU days just a few weeks ago. In here they used the system of Purkinje cells and the fact that they can actually change the amount of spike and also measure cerebral blood flow using imaging tools. So this is work that's been done in rats, if I remember correctly, and they're able to show first that you can find a clear relation between activity and cerebral blood flow, and that it's delayed. Cerebral blood flow arrives after activation, and also something that we also already know, that even when you remove the spiking, but you stimulate the area, but you remove you not allow output from the system, you will still get a cerebral blood flow change. This is not bold. This is cerebral blood flow measure within the tissue. Okay, different than what we did before. Before it was a bold measurement in fMRI. Here it's all invasive work, actually measuring change in the blood flow. Okay, so but it's actually resonant with what we saw before. There is a cerebral blood flow will be activated as function of stimulation. In this case, in the case of this area in the cerebellum, it's really sensitive to input more than the output, but you can abolish the output and you will still have a good activation in terms of cerebral blood flow, and it will be delayed as well. So much after the stimuli end, we still have a cerebral blood flow change that can be measured. So they use what did they use? They use, I think, fluorescent dyes that they put inside the blood vessel, and then they see a change in flow. They can see how much flow there is in the veins in their area of measurement. So they open. Because in fMRI it's like the correlate off, but they want to actually measure that's work. You want to, here's the work, this validation work. You want to check what is the blood flow exactly. You want to measure it, really measure flow. You put some dye and see how time it take it to move from one position to another. You can actually measure then time that it take it to flow. Okay, and you can relate that to activation that you directly do to this region. Maybe you put an electrode and activate the region and measure spike, or maybe blocking spike and still measure the change in flow versus this change in flow versus activation. So we're making this connection in more direct way. Right? Until now, I was saying that there is a connection. Now we actually see it in a measurement. We try to establish. Before it was not. I mean, the order of things was that this work starts earlier. This is the physiological people who were showing the relation between this blood vessel system and function. But just the idea that there is coupling is interesting and wasn't known in the beginning. It just became also very important for people who are measuring it with fMRI. So it started as a separate field, which there is not in there. People become very interesting in the beginning, kind of esoteric work, because it's kind of like explaining things about fMRI. There was another question? Okay. Next, and not only measuring, so besides measuring activity here, changing in, I think it's capacitance, if I remember correctly, but I'm actually not certain now, cerebral blood flow, amount of oxygen in the tissue, and use of oxygen in the tissue during activation. And what they found in this work is one that all those things, which we can imagine will affect our deoxyhemoglobin change, and they are not correlated. So they are correlated, but they are not one-to-one. There are these different functions that need to be described for each of them. all of them are in some delays, in different delays as well. Okay, so these different functions here, all of them are very slow, seconds, very slow, and are not exactly the same. So there is several mechanisms here that or changes in the that drive metabolism that will change, that in the end will change our measurement of BOLD. So that's kind of that's the important information that we should retrieve from this work. There's many more things there, but for us, that was also been shown with PET. PET, you remember we discussed PET. So in here they could measure glucose use versus oxygen use versus oxygen availability during seeing a visual stimuli, and blood flow. So they see a change in blood flow, change in glucose, which was kind of localized. That's important, right? Because it's localized. That was one thing that that was important for us. So it's localized. And those things change. Oxygen actually use wasn't changed so much, but oxygen availability changed a lot, which suggests that there is what? I kind of mentioned that in the very beginning of the class. In terms of oxygenation. Looking at this image, you expect what? When there is activity, there will be more deoxyhemoglobin or more oxyhemoglobin? More oxyhemoglobin. So kind of counterintuitive, but that's what we see here. Okay? And that's actually true. That's what we measure. That was many people found in the end. That's just saying that same thing. And so the people try to develop a model of the BOLD activity. And the thought is that there is a stimuli, then there is some neuron response, and then there's this machinery which we call hemodynamic response that derives this BOLD. This model called the balloon model, some of you might hear about. When people talk about fMRI, they talk sometimes about the balloon model. Okay, so like you blow into a balloon, use a stimuli that create this effect. Actually, afterwards, as we saw this, and it's important to notice that this function, that it's important. I wanna spend some time on it. There is this initial dip. There is a small reduction. That's actually the change in the oxydeoxyhemoglobin that happened just immediately after the activation. And this is a really good correlate of neuron activity. Just between very hard to impossible to measure. Okay, but exists. And then there is the secondary, if you want, effect that's coming several seconds afterwards. That's probably what we actually measure in fMRI. And then it takes very long time, about 20 seconds, to the system to go back and relax fully. Okay? And this thing, this hemodynamic response, or the BOLD response, is something that we use when we look on the stimuli. We're going to convolve this function with the stimuli and look for a signal which is a convolution between stimuli and the response. I'm not going to spend more time on what I just said. It's actually critical, but that's the basic analysis that's done in fMRI, and that's going to be part of what we're going to do in the next two classes. But just remember that came from here. So I took it from Leon Duell, which say, you see this thing. Some people see a hat, some others see an elephant, right? That it's like there's so many things inside it, right? That's the analogy that should come here. How it says in French? But it's important for you to remember that there are many sources of this BOLD effect. That's the main story here. Because the model then changed to add not only there is a change in cerebral blood flow that affects the cerebral blood volume, and something with oxygenation, which change oxygen-deoxy effect, so on. And then afterward, they kind of say, okay, all of those things are kind of dependent but separated. And if you really want to explain the BOLD effect, you need to account for all of them. That's kind of the measurement we saw in here. And most people are not doing any of that. They just say, okay, we have this function, a typical function, hemodynamic response. Let's convolve that with our signal and move on. Not discussing the fact that it might be different between regions, different between subjects. This is kind of like all gone. Okay, so what do you think is... okay, so we kind of think that this BOLD effect has to do with metabolism or energy consumption. And what do you think is the thing that actually costs in terms of energy in neural activity? What actually drives the signal? Why? Why there is this waste of energy, or there is metabolism and glucose is driving, oxygen is driving there? What actually driving it? What's so expensive in neuronal activity? The pump, that's good. Pump of what and what pumps?
Sodium potassium pump.
Okay, so say more.
And if you have a lot of input to balance to stay at the same volume as VR, the rest potential.
Okay, so keep the resting potential. The membrane to get back to resting potential, the membrane potential. To get to the to keep a membrane potential after depolarization or hyperpolarization, we need to use pumps to move ions against their gradients in order to be able to have another action potential, for instance, right? And that's what actually the thing that the most expensive thing in the system and drive most of the metabolism consumption. So that's what we see in here. So spiking restoration take about 50% of the energy use. Okay, this is in mice, by the way. Subterranean restoration another 34. So this is together. This is most of the energy consumption. There are other sources. It was interesting to see that there actually a difference between. And so that was the dominant view for many years. Now people replicate this work in monkeys, and there is actually a difference. For instance, where is a very impressive one is like how much action potential themselves affect in mice versus in monkeys, or the other way around, in mice versus in monkeys. So there is actually a difference, and so postsynaptic effect here it seems to be. So that kind of goes against what we said before. Here, at least in the monkey in this work, it seems that the postsynaptic has a big energy consumption effect, and that might actually. So that's kind of saying that maybe our signal is related to also to postsynaptic effect as well. It doesn't mean that it won't see it if we wanted postsynaptic effect. Even if we kill the postsynaptic effect, we still have bold effect. But the normal postsynaptic effect in monkeys, at least in this work, was highly sensitive to this energy consumption. Okay, postsynaptic energy consumption, and as we said, really moving ions from one side of the membrane to the other is the thing that takes a lot of energy. Okay, where is energy coming from? Does anyone have a thought? Do you remember anything from your biology early class? Huh? Yes, and there is something called what? Right, we create ATP, and it could go with anaerobic and aerobic cycles, right? And so typically we'll take glucose and break it. We have it here in. First, we can get two ATPs to get up to lactase, right? And then we can get much more using the Krebs cycle. Getting so this is the lactase, and then using the Krebs cycle, we can get many more ATPs, right? So the interesting story is, and one other important thing is that cells might have also glycogen, which is like this complex sugar, kind of that they kind of like thing they say within the cell, and when there is a need, they can move glycogen to, they can actually create from glycogen glucose and start this mechanism. The important story here is there is a difference between astrocyte and neurons. Neurons actually doesn't have any glycogen in them, and after activation, they just have no resource in the beginning. Might using this oxygen deoxyhemoglobin. They do this maybe very early change in this deoxyhemoglobin. They use from the environment the glucose, but then they have a shortage of glucose, and actually neurons cannot live in this situation. And so astrocyte, which does have glycogen in them, generate lactate and then release it to the extracellular matrix to be absorbed by neurons. Okay, so this is actually a nurturing system that astrocytes deliver lactate to neurons. Okay. More than that, this process is really sensitive to release of neurotransmitter, for instance, glutamate. So when glutamate is released, astrocyte will absorb it and start this mechanism which will release, which will then generate lactate that will be released for the neurons. Okay, so there is here coupling, and there are people who actually show. I removed that, but we can see actually activity related. We can see astrocyte activity which is related to stimuli, and in many cases, and you can think that has to do with that. They actually have to react really fast, so they're really part of the integral part of the system, because the neuron wouldn't act if you wouldn't have energy, right? So this system is a closed system, and when looking in, for instance, in the Allen Institute, we can find that this receptor of glutamate is really so. Do you know anything about this thing? Allen Brain Institute, and looking on you already learned about that. So when we compare metabolism, here we have a strong metabolism effect that they coincide with area which have a strong expression of this receptor. Okay. I think this is kind of saying the same thing. Yeah, neuron doesn't have glycogen, and there is this lactate that moves to the neurons. Okay, so now the system need to be. So first, okay, so this is kind of giving there is initial use, and the system react to it. That's the astrocyte neurons coupling. This now is coupled to the vessels, and that's typically done, it's very complex systems that include the smooth muscle that sit on vessels, and by open them and close them, you can change the flow to the tissue, flow of blood to tissue. So who control that? It's glia cells, but not only that interact with the vessels and kind of affecting the blood vessels. So now we have a system to change the flow as function of activation. So astrocytes that were creating a lactate for the energy consumption of the neurons will also interact, if you want, with the blood vessels, and also start this change in blood flow that will then change the oxygen, the oxyhemoglobin effect that we will measure. Okay, so this is the mechanism in the system. This is just saying the same. This system is super. How would I say it? It's a complex system. This neurovascular coupling. There are many neurotransmitters and ions so on that will affect this coupling between neuron, astrocytes, and blood vessels. So it's highly regulated. Many effectors that will change the blood flow. Here is a short list, or a list of some of the things so changing in pH, changing in potassium, changing in neurotransmitters that have been released in the area, and so on. All of them will derive change in flow that will then change the cerebral blood volume and the oxyhemoglobin and glucose amount in the region. One thing that need to be known, and is actually also relevant for us in terms of fMRI, this effect cannot be just local, but also have to, in order not to create really changing too much of big of a change in pressure in the tissue, there must be also an upstream. Information also has to go upstream along the blood vessels, not to create like just a big change in pressure and maybe hurt the blood vessels. So that kind of change a little bit the locality of the signal even further. Okay, this upstream effect. Okay. So this is basically it. This is the summary of this neurovascular coupling model. We're starting with synaptic activity, that then go with neurotransmitter release from neurons and which affect astrocytes that cause to vasodilation, and then also an upstream effect to avoid the effect of changing in pressure, and all of that will create the measured signal of POLD, the change in oxyhemoglobin that we are measuring with MRI. So it's kind of, for me, whenever I'm teaching that, it's such a complex, not direct measurement. It's kind of amazing that we're measuring with that in the end, function in the human brain. I'm actually, whenever I'm teaching, I say, really? And but it seems to work. It seems to work. Also seems to be understood now pretty well, and there is more and more evidence that that's the system, and it's something we can actually pick with MRI. Amazing as it is. Okay, I would just mention a little bit of consideration that we should have in the last 10 minutes that I have about blood vessels and stuff like that, and like things that happen currently, unless you have questions before I'm doing a shift. Okay, so blood vessels, size of blood vessels, and I mentioned that in the beginning. Big veins, big arteries can actually make a big effect. If tons of blood or tons of the oxyhemoglobin, change in flow there will be massive to our signal. They create a huge shift actually in the magnetic field around them. We can actually have people actually model it very nicely how and in what direction. They actually also have direction. So blood vessel would have a dephasing effect, which is in this direction from the blood vessel. So changing in orientation will change how the effect will go on the brain. And given that this is an MRI, a different MRI method, kind of very sensitive to T2 star, but a constant one, and you can see how these big blood vessels, which are kind of all around, right? And so if you look on in typical BOLD activation, you just look at the mean activation using a visual stimuli, okay? And this is visual stimuli, occipital cortex. You see here maps that people denote using fMRI, and you see that there is areas which are kind of like black. And if you look on shift in B0, so how much the magnetic field change, you can find like this area which is like V4. Is it like essentially there is no signal in here, and there is something here in the middle. So and that's a problem. You suddenly have areas which there is a huge magnetic field change, and so because there is a big, if you want T2 star effect, the mean signal there is just very low. There's almost no signal there, and that seems. So people actually noted that and looked at that. It took some time, and then they realized that. So when you look on this, so if you put the two maps, the one that we do to see blood vessels and this activation, you can actually find that there is this huge sinuses that goes, if you want, just through V1 and the others. Okay, so there are areas which you just cannot measure, or it's really hard to measure, and you always need to be aware about that. Also, there is some variation where they are in space. And here is again, the orientation actually make change of how they will affect the signal. So if you look at maps, and this is, it doesn't say anything to you right now, right? Only for some. This is fMRI map of eccentricity. So they show stimuli in different eccentricity. Okay, and then they project it on the brain. They do the same thing here with polar angle, and they project it on the brain. So with the polar angle, they actually they find maps. I mean, V1 is a full hemisphere, so it goes from red to green, and then the other one, let's say V2, I'm just now giving you a short talk about vision mapping and so on. But V2 will be just a quarter field. It goes from green to blue. Okay, and then it changes again from blue to green. That will be V3 and so on. Okay, so that's how we map things with fMRI. That's wonderful. And in here we see the eccentricity. We notice that there is a fovea. Things goes from red, from this red to purple. Okay, and that's also typically as it should be, as we know from recording and so on. Everything is great. But then we see this area which is like strange. Suddenly we have like the most far eccentricity just in the middle where phobia should be. And this is, so people are kind of like, I don't know what's going on. And afterwards they actually realize that that's exactly where I showed you before the brain goes through. Okay, so just be aware that's the wonderful thing. And then we also have issues sometimes. In terms of or something? No, it was in my head. Fine. I will just go fast now. Last few minutes. People now try to use graded field strength and different sequences. Try to go from what we had before with gradient echo to a different sequence like spin echo, which have much less signal, much less sensitive to the veins effect, but it seems to be more related to the capillary versus the bigger veins. And then people hope to be able to see better cortical structures. For instance, they can go now with 7 Tesla to very small voxel size. So I didn't tell you anything about voxel size in this class, but that's where we are right now. I mean, we started with PET, which was in the centimeter resolution. Then the early work in fMRI was in millimeter, but many millimeters here, 10 millimeter resolution voxels. And right now we are about here, so somewhere between 1 to 2 millimeter, typically 2 millimeter. This is the resolution used in fMRI. Still pretty big, but it's getting better. And what people are doing here now is try to go to even lower resolution using very high magnetic fields. Do you remember why high magnetic field? Because what else? There is a change in the SNR. The signal is greater as you go to a higher magnetic field. So we actually just have more signal in higher magnetic field. The gradient thing is very important. It's a different aspect. We use greater gradient to make this fast changing gradients. But higher magnetic field is in order to get better signal. Yes. But also you to do so you use this faster gradient in order to maybe break the volume to smaller voxels, but you could have not done it if you wouldn't have enough signal. And so that's why we're going to higher magnets. And there we're going to this relatively small resolution. People try to push toward the level of columns even. And here they kind of try to show you that they think they're able to see this ocular dominant column in the cortex. So that's where people now pushing fMRI. And really in the very, very last 2 minutes that I got, I will tell you about a different effort also done in 7 Tesla. So this also, instead of just measuring this contrast of T2 star, say we're not going to measure contrast, we're actually going to measure directly flow. We're going to kill the signal and then see how it goes back. That's it's a harder measurement, take longer, but they say it's a quantitative measurement of flow, which is closer to the cerebral blood flow that we saw before in mice and so on. So actually trying to do that in fMRI, that's a method that exists for a while, but nobody using it. So what is the benefit? And then what they did here, it's a very recent work, that's done again in 7 Tesla, and now they say, do you remember I showed you before that you know that there is a difference in terms of cellular distribution along cortical layers, right? So when you have very high resolution, you can start to create like these layers along the cortex. So it's not only that there is a change in cortical layers density of cells, there's also change in blood vessel along the cortical layers. And when they're measuring cerebral blood flow, so if you look on that, there is like, if you look on cerebral blood flow, there's these two main peaks in density of cells and then in vessels, and they would say that with cerebral blood flow along these columns, they have a better way to resolve this different layer, different layer activation with measuring the change in cerebral blood flow versus just measuring bold. And I would stop, but they show how beautiful and sensitive it is to different activations. So they would say this is the future. That might be the future. The problem in here is that it takes very long, but it actually gets you to this level of sensitivity that you would like to have. So that's nice. Okay, thank you. And you're really welcome to stay for the talk of Ariel.
אז יש שם חומר ממש טוב, תיקח את זה מחקר, נעשה את הכביש הזה, ויש גם חוקר כאילו למדתי על זה ככה.
טוב, אל תתחיל להשתלט לי עכשיו על הכל.
להשתלט לך על הכל. אז יש חוקר באר שבע שהמציא דרך להשפיע במקומות
אם לא, אני אדייק את השחור. מה אתה רוצה? אספרסו רגיל, כפול, סוכר, עניינים? רגיל. רגיל? קצר, אמריקנו? ארוך כאילו? אמריקנו. ארוך, יאללה.
חלב לא, חלב סויה, חלב פיזי. כן, זה עם הסרטים שלי.
יש לך וידאו שאתה צריך להקליט?
כן, עובד! בוא נראה הווידאו הזה עובר מקסימום אני אעבור אחר. אני יודע כבר מה לעשות. הנה, עובד. עובד גם עובד. נראה לי כל יום שלנו.
אוקיי, אז את זה קצת הצלחה, ומפעיל כשזה יתחיל.
אז אני אשים את זה פה, כדי שאני אזכור. זה אני שם.
כן.
אני מדבר בלי בכל דבר.
כן?
בעבר כבר פרשתי מיקרופונים.
כן?
לא, מה שעשיתי דיסטורשן בווידאו פעם.
ופה אתה מפעיל את זה פה.
שאני אעשה לך?
כן, לא רק לבדוק מנהל שומעים אותך.
שומעים אותי? זה אמור להגביר גם? זה מגביר אותי? אוקיי. שומעים אותי? יפה. אוקיי. אולי אני אחזור.
אתה רוצה להשתמש בלייב? בכל מקרה פה כמה דקות.
לא, אני מהצד של ההקלה. לא של ה...
יש איזה שקע שאני יכול לעשות?
כן, ממש פה למעלה. שהוא נאבק לך רק לפעמים.
תודה רבה.
מה העניין?
לא, מסתכל.
לדעתי זה להביא את זה, אנחנו מדברים ועשיתי את זה לדעת ויש פה פוינט, אתה רוצה פוינטר של האוויר? זה לא מעביר
מה רציתי להגיד לך? דבר אחד שיצא של המודל הזה, בבוקסים ושיפוע, צריך לעשות ספוט. מה שרוצים ב-MRI, לוקח מצבה, לוקח בוקסים מסביב קבוצה של מגביר את הבוקסים, המרכז, לספוט לעצים כזה.
ולמה זה אמור?
מאוד גסה, אבל היא פעם.
בלר, לבלוט, לעשות בלר.
כי כרגע, אם הבנתי נכון, הבעיה שאתה לא יכול למדוד בבוקס יחיד. אתה פעם, אתה צריך קבוצה שלך.
אתה מציע לעשות את זה פשוט באופן יותר אזור, או יותר קבוצה,
יש הרבה מאוד מקומות שבהם זה יעשה לך דברים.
מובינג...
כן, מין חסרון. זו חידה מעניינת שם, החידה הזאת, איך אתה עובר מהקבוצה לבוקס יחיד. זה בוקס יחיד, כמו שלך. גם זה בוקס יחיד.
אתה יודע, הכל בסדר?
כן.
שרון בחוץ, אתה רוצה? איך זה העניין שלך?
כן, מה שמע? מה קרה שקיטה?
זה כבר הרבה כתוב של
אולי נדביק אותה. בוא תדביק אותה.
אני אדביק אותך.
תדביק אותי.
אבל זה מה שעושה. תהיינו, נורא. מרגישה בידיים הייתי אולי טיפה מלוכדת.
אני אשאל אותך מה שאתה הולכת לקרוא.
קיצר, אני לא נהנית את זה, לא נהנית. זהו, לא באמת התחלתי, אבל אני עושה את עדיין לפעמים כזה שבוע הבא. זה קבעתי כבר כאן מחוץ, אני יודעת כבר, כי אני כאילו, כן, זה כמובן לוקח מלא זמן לכתוב מאמר, וכזה לפעמים זה ספר, כאילו בפועל אני כותבת אותו, אבל תכף מלא דברים שאני לא יכולים. זה יותר לי מוטיבציה לעשות את זה יותר.
זה יהיה מאוד
כן, כאילו מלא דברים כאלה בינורי, אבל זה הגיע למצב שפשוט כתבתי מחדש את זה. אני חושבת ברוב המקום יש כזה מקום לשים מי כתב, מי עשה כל דבר, כאילו, לא זה אפשר לעשות משהו של שני מילים.
אני לא יודעת, חשבתי שחשבתי על שנים.
כאילו, זה נראה לי לגיטימי, אני באמת לא מתכוונת. אבל כאילו, זה נופלים לי עליו. עשה מלא דברים. כן, אבל יש לי תוצאות לפעמים. זה מרגיש, אני לא ארגיש, כן, אפילו מקסימום אם שבוע הבא חלק מהיכולות, אז תדבק מחדש. או שזה ממש ציון ארוך. אני כבר מרגישה שאני מחלימה. נראה לי קראתי את זה, אני ממש חולה. ממש קראתי על זה יום השלישי. נו, ראית את המנה שלי? אמרו לי, זה ממש שאני כאילו שהצלתי אותם.